NeuroScience News Reading. NeuroScience News Reading. You're listening to NeuroScience News Reading, presented by Hakeem Ali-Bocas Alexander for World Reading Club on the Wisdom app in association with UniquilibriuM. Today's reading is titled, How Many Drinks is Too Many? How many drinks is too many? I'll start off by reading from NeuroScienceNews.com. NeuroScienceNews.com, How Many Drinks is Too Many? Under the category of Featured Neuroscience and Psychology, published today, September 12th, 2022. Summary. Even low doses of alcohol can spark transcriptomic and epigenomic changes in brain areas associated with addiction. Source: University of Illinois. A new rodent study shows that even small quantities of alcohol can trigger epigenomic and transcriptomic changes in brain circuitry in an area that is crucial in the development of addiction. What's more, the University of Illinois Chicago researchers who conducted the study say that the pathways involved in priming the brain for addiction are the same ones that are associated with the highs of drinking, like euphoria and anxiolysis, the clinical term for a level of sedation in which a person is relaxed but awake. This suggests that when the brain experiences the anti-anxiety effects of alcohol and the mood lift, the relaxation and the buzz, it is also being primed for alcohol use disorder, said the study's senior author Subhash Pandey, the Joseph A. Flaherty Endowed Professor of Psychiatry and the director of the Center for Alcohol Research in Epigenetics in the UIC College of Medicine. While Pandey says the study does not suggest, for example, that one drink leads to addiction in humans, it does provide some clues as to why some people are more vulnerable to alcohol use disorder. We're seeing that dependent behaviors may not always be from long-term, high-quantity habits, but a result of rapid epigenetic changes in the brain, which we show in this study may start happening even at low doses, said Pandey, who is also a senior research career scientist at the Jesse Brown Veterans Affairs Medical Center. A paper published in Molecular Psychiatry details Pandey's experiments, which studied rats under control and alcohol exposure conditions. So, a paper published in Molecular Psychiatry details Pandey's experiments, which studied rats under control and alcohol exposure conditions. You're listening to NeuroScience News Reading, presented by yours truly, Hakeem Ali-Bocas Alexander, here on the Wisdom app for World Reading Club in association with UniquilibriuM. NeuroScience News continues. In the experiments, rodents were exposed to low concentrations of alcohol, and their behaviors were observed as they explored a maze. Then the researchers analyzed brain tissue samples collected after euthanasia with RNA sequencing and looked for gene expression patterns. When they analyzed the samples, the researchers found that a gene called hypoxia-inducible factor 3 alpha subunit, high F3A for short, was associated with changes in the brain after alcohol exposure and behaviors, like how long rats stayed in areas of the maze with enclosed, high anxiety, or open arms, low anxiety. Alcohol increased high F3A expression even after low doses of exposure and reduced anxiety. Note, I'm not sure if this is pronounced in the scientific community as HIF3A or high F3A. I'm choosing to pronounce it high F3A. NeuroScience News continues. And while many effects of alcohol are different among males and females, there was no difference between the two in this study. We saw that low doses, what we consider social drinking, changes the gene expression in the amygdala, a brain region that regulates anxiety. In other words, it creates an epigenetic pathway for addiction, Pandey said. Pandey and his colleagues also set up additional experiments in which they blocked the gene in the amygdala of rats with or without alcohol exposure to validate its role mediating anxiety. Anxiety. When high F3A was blocked, anxiety was increased in control rats, mimicking withdrawal from chronic alcohol exposure. On the other hand, this also prevented the anti-anxiety effects of alcohol. The researchers showed why, too. High F3A's chromatin, bundles of DNA and RNA, is loosely bundled, meaning the genes are easily accessible for transcription changes. One thing the study does not suggest, however, is what level of alcohol exposure was safe for rodents. Instead, Pandey said, it's important to know that low doses created a priming for addiction. For people, he thinks the takeaway is simple. Don't assume social drinking or even pandemic drinking is without risk. Technology. You're listening to presented by Hakeem Ali-Bocas Alexander on the Wisdom app for World Reading Club in association with UniquilibriuM. NeuroScience News continues. Alcohol use disorder is complex and challenging to overcome. The information we learned from this study helps us to understand better what is happening in the brain and, one day, may be leveraged to develop better treatments and pharmaceuticals, Pandey said. Co-authors of the study, titled Unraveling the Epigenomic and Transcriptomic Interplay During Alcohol-Induced Anxiolysis, are Harish Krishnan, Huibo Zhang, Ying Chen, John Peyton Bonsack, Annie Xie, Handojo Kusumo, Jenny Drenevich, Chunyu Liu, Dennis Grayson, and Mark Meinschein Klein. Funding. The research was supported by grants from the National Institute on Alcohol Abuse and Alcoholism and the U.S. Department of Veterans Affairs. NeuroScience News suggests you see also from June 3rd, 2022, how wisdom, resilience, and mastery work together to boost well-being in old age. Under the categories of feature, neuroscience, and psychology. About this neuroscience and addiction research news. Author, Jacqueline Carey. Source, University of Illinois. Contact, Jacqueline Carey, University of Illinois. If you're looking at this on neurosciencenews.com, the image that's used here is in the public domain. Original research, open access, unraveling the epigenomic and transcriptomic interplay during alcohol-induced anxiolysis by Harish Krishnan and others in molecular psychiatry. How many drinks is too many? How many drinks do you think is too many? Continuing. NeuroScience News continues with an abstract from Unraveling the Epigenomic and Transcriptomic Interplay During Alcohol-Induced Anxiolysis. An abstract from Unraveling the Epigenomic and Transcriptomic Interplay During Alcohol-Induced Anxiolysis. Positive effects of alcohol drinking, such as anxiolysis and euphoria, appear to be a crucial factor in the initiation and maintenance of alcohol use disorder, AUD. However, the mechanisms that lead from chromatin reorganization to transcriptomic changes after acute ethanol exposure remain unknown. Here, we used assay for transposase accessible chromatin followed by high-throughput sequencing, ATAC sequencing, and RNA sequencing to investigate epigenomic and transcriptomic changes that underlie anxiolytic effects of acute ethanol using an animal model. Analysis of ATAC sequencing data revealed an overall open and permissive chromatin state that was associated with transcriptomic changes in the amygdala after acute ethanol exposure. We identified a candidate gene, high F3A, also known as hypoxia-inducible factor 3 alpha subunit, that had open chromatin regions, ATAC sequence peaks, associated with significantly increased active epigenetic histone acetylation marks and decreased DNA methylation at these regions. The mRNA levels of high F3A were increased by acute ethanol exposure, but decreased in the amygdala during withdrawal after chronic ethanol exposure. Knockdown of high F3A expression in the central nucleus of amygdala attenuated Knockdown of high F3A expression in the central nucleus of amygdala attenuated acute ethanol-induced increases in high F3A mRNA levels and blocked anxiolysis in rats. These data indicate that chromatin accessibility and transcriptomic signatures in the amygdala after acute ethanol exposure underlie anxiolysis and possibly prime the chromatin for the development of alcohol use disorder. NeuroscienceNews.com asks you to join our newsletter. Sign up to receive our recent neuroscience headlines and summaries sent to your email once a day, totally free. We hate spam and only use your email to contact you about newsletters. You can cancel your subscription anytime. NeuroScience News posts science research news from labs, universities, hospitals, and news departments around the world. Science articles can cover neuroscience, psychology, AI, robotics, neurology, brain cancer, mental health, machine learning, autism, Parkinson's, Alzheimer's, brain research, depression, and other topics related to cognitive sciences. You've been listening to a reading of How Many Drinks is Too Many. This is NeuroScience News Reading, presented by Hakeem, Ali-Bocas, Alexander, here on the Wisdom app for World Reading Club in association with UniquilibriuM. How many drinks is too many?